this intro, we could say like the cell cycle is a process by which somatic cells replicate. This happens in 3 stages: interphase, mitosis, and cytokinesis, and then so on, so on, so on, just explaining what the cell cycle is. So this is important because it gives a background to the reader. You kind of have to assume that the person reading your lab is not going to be that knowledgeable. So this is the purpose of the introduction and what the introduction is. So moving on, the purpose. So the purpose of the experiment is kind of saying, like, what are we trying to do in this experiment? Why are we doing this experiment? So again, going off that cell cycle example, you could just say the purpose of this experiment was to see which proteins inhibit the cell cycle. If the lab that we did or experiment we did was we were looking at proteins that inhibit the cell cycle. So you should always use a purpose because it explains to the reader, what are you actually doing in this experiment? What goal are we trying to achieve? Okay. So next is your hypothesis. So I think all of us know what a hypothesis is just like, I guess you could say your opinion on what is going to happen in the experiment and what the results are going to look like. So, a lot of you probably have learned that if-then hypothesis, like if this happens, then this happens. You don't really need that. A hypothesis can be a format. I wrote my example one here. The p53 protein will inhibit cell division. The RAS protein will promote cell division, and the p21 protein will inhibit cell division. So as you can see, it's just a general opinion on what we think is going to happen. And the real purpose of the hypothesis is that usually when you do a lab or an experiment, your teacher will give you like some background reading. So maybe they'll explain what each of these proteins is. So then from that, you will make an inference based on, any prior knowledge that you have. So that's the purpose of the hypothesis. Okay, so next we have variables. Now variables are like really, 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 really important. And there's 3 types. You have the dependent variables, independent variables, and then you have your control. So your dependent variable, a good way to remember the difference between dependent and independent is that dependent depends on the independent. So pretty much like in our experiment with the proteins and the cell cycle, we're going to be changing, or what the dependent one is like if the cell cycle occurs or not, because we're looking at what effect do proteins have on the cell cycle. So the proteins have an effect on the cell cycle, which means that the cell cycle is dependent and then the change in proteins is independent. Independent is always what the scientists change, and your dependent is you're going to be your data. So try to remember that. That's really, really important. So now, I didn't write this here, but your independent variable, you're going to have multiple groups. That's what like creates the different groups. So like, in this experiment, right, each different protein, like p53, p21, and RAS, those 3 different proteins in this, going off of like the cell cycle example, each of those different proteins is going to be kind of like a different group in the experiment. So like the p53 will be one experimental group, p21 will be one, and then RAS will be another. But you also need to have a control group. Now, we discussed this like in our first class when we discussed like experimental method. But like your control group is kind of like the baseline that you're going to be looking at. So the control group kind of tests, make sure that there's no flaws in your experiment. So for our example, you might want to, just run the experiment with no proteins to see if your cells actually go through the cell cycle. Because let's say, the water is boiling, then your cells will probably not go through the cell cycle and break apart. That's why it's really important to have a control group, because it makes sure that your experiment is kind of working properly and there's no flaws in your experiment. So those are kind of subsets of the independent variable, right? You're going to have the control group and your experimental groups. Now you also have the control variables. Now these are variables that do not change. And it's very, very important that you mention these, because if you have two independent variables or two variables that are changing, that means that or whatever effect it has on the experiment, it's hard to measure what variable is causing what. This is why you need control variables. So for example, in our experiment, our control could be the concentration of the protein. So if we keep this standard throughout all of the proteins, then we know that, if one protein is in like really, really high concentration, it's not going to have weird effects than if it was like in a really, really low concentration compared to another. So these are things you just want to keep standard to make sure that, they won't affect the outcome of your experiment. Those are control variables. Another really, really important thing that I can't stress enough is that your variables must include units if you're using them. So for example, if you're going to measure temperature, make sure you put that your dependent variable will be in Fahrenheit or your variable will be in Celsius. If you're measuring volume, make sure that you write down that it's going to be measured in milliliters, or it's going to be or it's going to be measured in liters. This is really, really, really important. Make sure you include units. Um, in this case, in our experiment, so the dependent variable, if the cell cycle is occurring, I mean, that doesn't really need units because it's kind of just like a yes or a no question. The independent variable is which protein is in the solution. Again, you're not going to really need units for that because it's just like different proteins. But for the control group here, concentration of protein, you're going to need a unit for that. So like moles per liter will be a concentration unit. So make sure you include units in your variables. Okay, so next is our materials and procedure. So materials, you kind of just want to list everything you used. For example, if you used a beaker with like 500 mils, if you use thermometer, if you use a magnifying glass, use a glass slide, just list it. Make sure that you're listing all the materials that you used. And then you have the procedure, right? So the procedure is very, very important in lab reports because it kind of gives the teacher as well as other people who might try to replicate your experiment an understanding of what you actually did in the experiment. So in our example, I only wrote like the first two steps. So one for the first step could be mix the protein solution that we were given with 500 milliliters of water. And then the next step could be do this for all three proteins. So you want to like make your procedure pretty detailed. Make sure that you're kind of just going over every step you took to do this experiment. You can always, use like quick annotations, right? Like I didn't write mix protein or the RAS protein solution with 500 milliliters water. Mix the P53 protein with 500 milliliters water. I just kind of said do this for all three proteins. So that's another way, you can just make your procedure shorter, but you're still including all the steps. Make sure you include all of your steps in your procedure. You do not want to skip anything. Usually procedures will be like 10 to 15 steps long. I mean, if you have like a really complex experiment, then it'll be more than that, but usually it's only 10 to 15 steps long. So yeah, that's pretty much it. Okay, so the next thing is data, right? So data can be in many forms. It can be like a temperature reading. It can be any kind of measurement. It can be objective measurement. So just when you're using or when you're trying to visualize data, you always use a graph or a table. So usually the data section of your lab report will just be a graph or table. Nothing more, nothing less. It's pretty simple. I mean, you just kind of plot all of your data points, make sure your trend that you're getting is what you expected, if it's not what you expected. And okay, one important thing about data is that graphs and tables, any kind of visual thing, should be titled. Also, if you're using a graph or even a table, your X and Y axis should have variables as well as titles. So this is really, really important because you don't want to just include almost like like just some values on a table, it kind of doesn't mean anything unless you title the entire table or graph as well as the X and Y axis for each side. This way it actually makes your data understandable. And you don't need to include both the graph or table, just include one with your data values unless you're given specific instructions. So all of this that I'm telling you is just kind of like a general thing that you guys need to know. Remember that like a lot of teachers have different views on how lab reports should be written, what should be included in each section, what needs to be included, and what does not need to be included. So it's very individualized per teacher. So in that sense, you guys just listen to your teacher and they'll tell you everything you need to do. And so that was data. The final thing is analysis and conclusion. So this is kind of like the hardest part of the lab report because it kind of requires your own thinking. So the first thing that you want to do in an analysis or conclusion is always compare what you got in your results to your hypothesis. The reason you do this is because it's kind of like almost, it will act as like an introduction for your, I guess you could say, for your experiment, for your conclusion, and just explain if your hypothesis was correct or not. And remember to explain why. The analysis inclusion and conclusion is pretty much made up of a bunch of claims that you have, and then you just have to explain why. If you do that, then you'll be fine with your conclusion. Explain why the results were the way they were, and then provide more insight on them, maybe some background knowledge that you have that you read up on that will continue to support the evidence that you found in your experiment. Or if you got some completely new evidence, then maybe you'll have to do some research, figure out why you got that evidence, and then you can write that in your conclusion. A lot of times teachers will give you conclusion questions to promote your thinking. They'll ask you, why did this happen? Why did your results come out this way? Or so on. And they'll kind of do the hard work for you, and you just have to answer their questions. So that's it for analysis and conclusion. One more thing though, a lot of times, lab reports, sometimes teachers would kind of just give you a packet with a bunch of questions, and then you just kind of fill out everything. But it'll make you do all of these steps, right? So like they'll ask you, maybe they'll give you like a background sheet, and then you have to do some like multiple choice questions on them. That'll be your intro. Then they'll make you explain, what are we doing this experiment? That would be your purpose. Then they'll say, what do you predict will happen with your hypothesis? They'll ask you for like your control group, your variables. Then they'll ask you, what are we using in this experiment? How are you going to conduct this experiment? And so on. The really important thing about labs is you should always follow instructions. I know it like seems obvious, but like usually teachers will give you like a set of instructions. If you're unsure about any steps, don't hesitate to ask, because oftentimes you have a limited amount of time to conduct these experiments. And if you spend more time worrying about if I'm doing the right steps, you don't actually get the experiment done, and then you can't really write a report. So make sure if you have any questions, just always make sure you ask, because it'll really, really help you and make sure you don't make any mistakes. Oftentimes teachers will give like confusing lab reports or labs just because, sometimes the instructions will be hard to follow. And oftentimes the labs are the hardest part of biology because, biology is kind of just like any other course like history or English, a lot of memorization and a lot of memorization concepts. But the different thing is that you have labs, and those are very, very hands-on activities. So you're not just sitting at a desk memorizing stuff, you're kind of actually doing stuff with people. So yeah, that's all I really wanted to cover for this class. That's really it for this entire course. We've kind of covered everything. We've covered, all of the major topics that you will need for high school biology. If you guys have any questions on anything in biology when you're doing it this year or next year or so on, you can always just email me. I'm pretty active, and I'll just get back to your questions. I mean, that's pretty much it. If there are no questions on this material or any material we covered throughout the year, then we will end here. Okay, well, in that case, we've finished this course. You guys have all finished the course. Congratulations, you now know high school biology. Good luck in the upcoming school year. If you're taking biology or not. If you're taking biology, well then I would recommend, just going over the lectures or, going over any set of biology lectures to refresh your knowledge, what we did in this class or, what you learned. If you're not taking it, well, our biology this year, you can still just review some of the lectures, just refresh yourself again. And with that, thank you all for attending this class, this series. And yeah, if you have like any feedback, I will be posting a form later today with a feedback section. And if you guys want to see me teach any other classes or anything like that, you can also recommend that there. So yeah, thank you all for attending, and yeah.